Come on, chat. Perfect. So yeah, so today, like I said, we're going to focus on chapter 1. And so this chapter basically just talks about how diverse the cells are in our environment. They talk about how we look at cells under the microscope, different types of microscopies that are available. We're going to be reviewing prokaryotic versus eukaryotic cells. We're going to talk about different parts of cell structure and a little bit about its function as well, although we're going to go through that in a lot more detail as the semester progresses. And we're going to end with model organisms, things that we use, actual organisms that we use to study cell and molecular biology of these cells. So that is our basic information. So to start with, when we look at cells, right? Well, when we look at our environment, we see a lot of different types of organisms. Even two humans don't really look alike, right? You have a lot of variety of various animals and plants and bacteria and viruses. Little thing doing all the work that it needs to do. Okay, so I have 12 things in chat. What happened? Did I freeze up?
Yeah, the video is just freezing.
Okay, am I unfrozen right now?
Yeah, we can hear you.
Yeah. Yes.
Okay, I'm gonna switch my internet if you wouldn't mind. Give me a second. Okay, hopefully this will be better. Okay, so if we look at cells of various types, we're going to see that they're going to vary in their appearance and in their function. So we talked about, we were talking about these cells when we're looking at a single cell organism, right? A single cell that can just lives a solitary life, right? It's just does its own thing. It's, you would say, well, it's going to be a very simple organism, but it's not. Depending upon what its function is and what type of environment it lives in, it can vary a lot, even in that single cell organism state, right? Some of them have long flagellas, some of them have pseudopods that move around, right? Help it crawl around here and there. Some have cilia, like over here, you see a cell covered in these little hair-like projections that help it move through its environment. So they are going to be very different simply because of the environment they live in or the type of things that they are surrounded by. Also, what can affect their appearance could be their ultimate function. So if you were to look at a neuron right here, you can see my cursor moving around, guys?
Yeah.
Yes?
Yeah.
Yes.
But yeah, so if you look at a neuron looks very, very different. It almost looks like an upside down tree in this particular image, but they have a very small cell body, but then they have this long axon to help it communicate, right, with other cells far away from itself. And it has tiny dendrites all around it, these little projections that kind of look like root hairs or tree branches that help it interact with other cells around it in its immediate environment. On the other hand, if you look at some plant cells, they can appear almost immobile or inanimate, right? They appear like they're not even living in that image because of the way they look and behave. So these are very boxy cells, right? Because of the presence of cell wall around them. Many times, if they're on the surface, they may be coated in a wax-like coating that gives them even more inanimate appearance. And here you have a yeast cell that's in the middle of cell division, binary fission of some sort, so that it is dividing into two daughter cells. Now, just because the cells vary so much in their appearance doesn't mean that they cannot still share some important components inside them about the way they interact with the environment or the requirements that they have in order to be called living cells. So the cell theory basically talks about how all organisms, anything that has come to be living in this environment on Earth, is composed of either a single cell or multiple cells, and that all of these cells have come from some single source at some point in the history. So they have all come from some pre-existing cells. They didn't just come about on their own randomly everywhere, but rather there was a single cell somewhere that first came, and then somehow through interaction with its environment, through changes slow over the period of time, has converged into all these different types of animals and plants that you see in the world today. Now, to start with similarities between all these organisms, whether they are a single cell, a multi-cell, or a plant or an animal, we go inward into the molecules. And we find that one, they're all going to be organic molecules, which is what we're going to focus on in chapter 2, but also that they all basically have the same chemistry. So they all have an instruction code inside them, either in the form of DNA or RNA, that's made up of nucleotides. And that instruction code is used to create worker molecules in the form of proteins that are composed of individual units called amino acids. And those proteins are then what makes us who we are, makes us look the way we are, makes us work the way we do, and do the perform the functions that we do. Now, how did we learn about all this stuff? It has been through centuries of research and centuries of slow development initially, and then fast development in the last couple hundred years of materials that were used to study these cells at a microscopic level. So before the invention of microscopes, it was very hard to figure out what life was made of, or if there were any similarities between all these variable, organisms that were present on Earth. But the invention of the light microscope back in the 17th century led to the discovery that all living organisms, all living beings seem to be made of similar structures called cells. And then later on, as this slowly developed and we were able to develop stronger microscopes to look at internal structures, we were able to reveal some of the cell components, look at the nucleus, look at the other components that were present there. Now, we can do a lot of cool things with a light microscope, as you will see in lab as well. However, light beams can only resolve substances to a certain, degree. They cannot really be used to look at the tiny, tiny organelles that are present inside the cell. To do that, to look at those fine molecules inside the cell, to look at the organelles inside the cell, we had to develop something more, advanced, something that could resolve smaller objects. And to do that, what we have now is electron microscopy. In electron microscopy, instead of using a beam of light as we do in a light microscope, instead of that, we use a beam of electrons. So it is able to resolve to a higher degree those small structures, and we can look at structures that are 20 nanometers or even smaller with that. On top of that, we've had a lot of other modern enhancements to extend the functionality both of light microscopy as well as electron microscopy. Some of the ones that are specific for light microscopy are being able to look at different phases of the cell, so-called phase contrast, to look at the various structures inside. To be able to employ fluorescence, so we can make fluorescence probes to tag specific parts of the cell and then examine those. To look at dark field or to look at confocal, where we can create a 3D structure of the cell. So when we're looking at cells under a microscope, how big are these cells exactly? This kind of gives you a little bit of that information. So if we were to look at our cells, the cells, some of them can be in our visible range so that we can actually see them. An example of that would be a chicken egg, right? It's a single cell. We can see it with our naked eye. There are other cells like that that we can see with our naked eye as well. Typical cells, eukaryotic cells range from 2 to 20, sometimes even larger micrometer. However, bacteria and some of the other cells like that are going to be very small. They're going to be way smaller than that. The organelles inside the cell, on the other hand, are in the nanometer range. So they could be 20 nanometer to like 2 micrometer, depending upon what it is you're looking at. A single ribosome that is used to make your protein is usually about 20 nanometers, so extremely small. The plasma membrane around the cell is 5 nanometers, so even thinner, right? Even smaller. The atoms, on the other hand, are going to be not even a nanometer long. So the microscopy techniques that we have, no matter how advanced they are, will not be able to let us look at these small atoms right now to the same degree as we would like to, right? We can't see a single oxygen atom walking around in there right now. We can see some larger molecules, however, with the aid of the electron microscope. The light microscopes are going to be best to work with these cells in general, just single cells. Some organelles can be seen with that as well, but they are still going to be no more than, within several hundred nanometer would be their limit. Some of the super resolution fluorescence microscopes we have available today can get a little bit better. They can resolve some of those smaller structures. So we can see some of the DNA strands a little bit more in detail. We can see maybe some ribosomes in some of the cases as well. However, to look at anything smaller than 20 nanometer, the best thing to use is the electron microscope. So this is going to go through that a little bit more in detail. So in light microscopes, basically what we're doing is the light microscope, like the ones you will use in the labs, are what we call compound microscopes. They usually have 2 sets of lenses. So we have our object on the stage. We first magnify it once with the help of our objective lenses, with the help of our ocular lenses, and then a second time using the objective lenses. And by taking that magnified image and magnifying it further. By doing that, we can usually resolve the cells up to 1,000-fold. So anywhere from 40x all the way up to 1,000-fold. Now, if we were looking at these cells just by themselves, the way they normally occur, it's hard to see their structures because everything is kind of just clear in many cases, unless you're looking at chloroplasts, which will be green. But a lot of these structures are uncolored, so you are not looking at a lot of differences in them. However, there are differences in the refractive index of the various organelles that are present inside. So if you use a phase contrast microscope or other techniques like that, you can resolve some of those systems a little bit better. A better way to look at them, however, is to stain them. And we do that by using several types of dyes that are available in the market, both just colorimetric dyes as well as fluorescent dyes that can color different portions of cells differently. So in this case, this is eosin and hematoxylin staining. You have the pink in the cytoplasm and the blue are your nuclei within this structure. This is probably a section of a tissue and you're looking at those cells. Say whether it is a plant or a human tissue or animal cell tissue, it would be the same thing. You kind of section them into small, tiny, thin layers, you stain them, and then you can look at them. Now you could do that by keeping the cell still alive, where you can still see movement of different parts inside the cell. But many times the procedures we use to either stain them or section them end up killing the cells. So in that case, you are not going to be seeing actual cells moving and living and dividing, but you will be able to get a snapshot of what was happening in that particular system at the time that you sectioned it. Here's another example where you can clearly see your, in this case, the cell wall on the outside, and then your cytoplasm inside in the pink area. So like I said, another way that we can look at these is by doing phase contrast microscopy, where we are able to work with the resolution of the cells in a way that we can look at the contrast between the nucleus and the cytoplasm, since they have slightly different refractive indices. So here in the center is your nucleus, outside is your cell membrane, and then inside this kind of blurry stuff is your cytoplasm. You also see some types of organelles that are around, right? You see some structures that are forming, but it is hard to figure out what they will be exactly. Some of this would be parts of the Golgi complex or the endoplasmic reticulum. Some of these might be some other organelles, but it is hard to resolve them using just the phase contrast and light microscopy. To resolve some of these structures a little better, we use those fluorescence microscopy, where we can use fluorescent dyes that are either tagged to an antibody to a protein of interest, or that specifically binds to a particular structure. Like we can use DAPI that stains the DNA, or propydium iodide that specifically stains the DNA to look at the nucleus, right? In this case, you have an antibody that is staining the cytoskeleton structure, right? Actin and the microfilaments in the red and the greens. And you can look at then the cytoplasmic structure and the structure of the cell, the membranes, with the blue nuclei inside that contains the DNA. So this helps us extend our light microscopy capabilities to make them more specific, so that we can look at some of the cells in more detail. Nowadays, there is a super resolution microscopy available as well that allows us to look at even more details inside the cell, as you will see. Now, another way that we can do microscopy in our lab to look at structures, these are more 3D structures in a multicellular organism or in a eukaryotic cell, is confocal microscopy. In confocal microscopy, you are using laser beams to go at various depths inside that cell and get an image at that depth. And then it kind of stacks them on top of each other to create your 3D structure. So if I was looking at this particular portion of this organism under, or tissue under a typical fluorescent microscope, this is what I would see. However, if I'm able to use confocal microscopy, I can resolve it to see the details inside that structure. I have links to other sites where you can look at 3D structures of various organs and various tissues using these microscopy techniques. So finally, you have, like I said, this new technique called super resolution fluorescence microscopy. It doesn't have to be fluorescence, actually. There is a microscope that is just becoming available in the market this year. We had the luxury of using it at UNF. We had it on loan at UNF last couple of years for testing purposes, where you can look at cells over time, just culturing as they're going. It creates a holographic image of the cells, and it resolves them to great degree, where you can see movement of molecules from one place to another. You can track the cell movements as they go from one place to another as well. In super resolution fluorescence microscopy, it uses the conventional fluorescence microscope, but then it extends it to, it kind of combines it with a confocal microscopy to create super resolved images in a way that you can see them in their actual size rather than blurry tubes as you would have seen before. And this is the difference in that structure. There are a couple of things in the chat. The chat is not visible to me right now. I'll figure it out before the next time. Could you please let me know what your question is?
They just wanted to know, like, if we still have that microscope available at UNF.
Oh, okay. No, we don't have the microscope available at UNF anymore. I do have actual videos and data from that I will share in the lab with you guys a little bit later in the semester when we are working with cell differentiation and the cancer biology module. So you will get to see some stuff from there. That microscope, the new microscopy method that they have developed, it's called Q-phase microscopy. And it, like I said, creates a holographic image of your cells and follows their, culture and movement over time. Okay, so finally, sometimes, so that's all good. We are looking at molecular things. We're looking at molecular structures. That's what we want to look at. But we also want to see microscopic organisms. We want to see actual virus structure or a bacteria structure that is not visible with the naked eye. And if you wanted to do that, many times light microscopy can't help you resolve the image enough to know exactly what's in that surface. It's just going to give you an outline, right? Just a bare outline. To do that, what we use is a special type of electron microscopy called scanning electron microscopy. So when we do electron microscopy, like I said, the biggest difference between light microscopy and electron microscopy is that in light microscopy, you are throwing a beam of light at at your tissue or at your slide. In electron microscopy, you're throwing a beam of electrons. Now, within those, within the category of electron microscopy, there are two different ways you can do it. You can do transmission electron microscopy or scanning electron microscopy. In transmission electron microscopy, it is a beam of electrons. It's looking at its structures inside the cell usually. In scanning electron microscopy, it's scattering essentially a bunch of electrons that are bouncing off and giving you the structure on the outside of that cell. So this way you can create a 3D structure of whatever it is you're looking at, whether it's a chemical compound, a ribosome, a cell, a virus, a eukaryotic cell, a bacteria, a virus, whatever. So here you can see two different images of that sort. On the left side is a human neuroblastoma cells. These are brain tumor cells that are getting eaten up by macrophages. So our immune system is recognizing them as something foreign and trying to kill them. And on the right side is a pollen grain. Now, not all pollen grains look the same. I had a research student a long time ago that was doing a project on antibacterial and antimicrobial properties of honey. And they had honeys from various locations across the country. And depending on what environment the honey was produced in, what flowers and what plants they were around, the pollen grains were different that could be found inside that honey. And those pollen grains looked amazing. They look very, very different. They are as variable in structures as our snowflakes, essentially. But we could identify the type of plants that the honeybees use to create the honey that we were collecting and then looking at. I might share some of those images with you as well, if I remember. Now, again, if we were to look at, this is the other type of electron microscopy, which is transmission electron microscopy. In this case, you're not going to get the 3D structure. It's going to be flat surface because this is just looking at one plane, looking at the cell's internal structure. So you can use it to look at cells' internal structure in a great detail. You can see individual ribosomes hanging out on the mRNA strand, creating protein. You can see DNA strand getting replicated. And you can see mitochondria. You can see all the internal structures. So this is a great way to see the insides of the cells. Okay, any questions up till here? Or a couple in the chat, maybe? I have to find the chat. It runs away. Do we still have that? Okay, that was still this. Okay. Oh, there it is. Yes, I got the chat on top. Life is good. Hopefully it will stay there. Okay, so next thing we're going to talk about now that we've looked inside the cell is the various types of life that's present and what are the differences between the cells that come in each one of those domains of life. So if you look at all the organisms, all the life that is present on Earth, they can be divided into two main categories first, prokaryotes versus eukaryotes. Prokaryotes are simply, if you translate it, it simply means before the nucleus or before that central structure. And eukaryotes is where you have a true nucleus. Within the prokaryotes, there are also two different domains. You have bacteria, which is what we typically learn about and interact with a lot more, and then archaea, which are even, when we compare them, they are as different from bacteria as bacteria are from eukaryotes. So they are their own little class. Archaeas are more adapted to really harsh environment and probably came first. Those were the more primitive organisms that were present here because they can survive without, many can survive without oxygen. They can survive in hot springs and harsh acidic environments, all kinds of really crazy environments that normal organisms don't typically survive. And then you have your bacteria and eukaryotes that are your other two main categories. Okay, so there are some things that are going to be common in all cells, regardless of whether they are prokaryotes or eukaryotes. And those things are going to be your cell membranes, which are the lipid bilayers that are surrounding the cells, and they are going to be essentially the boundary, right? That's the wall around the cell, or not a wall, but a boundary around the cell telling this is the cell. And then the cytoplasm, which is the aqueous solution inside the cell that all the other stuff is going to be housed inside. That aqueous solution inside the cell membrane, it's a jelly-like material. It's water-based, as you can see, aqueous solution. And it contains a lot of macromolecules, salts and sugars and things, in addition to the organelles that are going to be present there or the material that's going to be present there. Now, cells are going to be categorized either as prokaryotes or eukaryotes. And given an image, you should be able to define which one of those two categories it belongs in. The eukaryotes are going to have the DNA, your genetic information that tells all the instructions to make you who you are, inside a central nucleus that contains it, right? So it has its own little membrane. It's like, special section of the library. And the prokaryotes, on the other hand, are not going to have a nucleus. That doesn't mean that the genomic information or the genetic code is just going to be like everywhere. It's still huddled together, but it's not going to have a membrane around it to keep it inside. It's not in an enclosed space, but it is still intact. It's still together. Prokaryotes are going to have a lot more simpler structures. They're typically a lot smaller than your eukaryotes. They can live in a variety of environments. Depending, like I said, the archaeas can live in the harshest environments you can think of. But they still have a lot of variety. Just because they're single cells or they are simpler doesn't mean that they all look the same. They have a large variety of shapes and sizes that come within that category as well. They don't have any membrane-bound structures that we call organelles, but they do have molecules that do specific functions inside that their membrane. Some prokaryotes you can look at, like we said, we talked about, there are two domains, bacteria and archaea. These are some common shapes that you can see within the prokaryotic world. These are all examples of bacteria. So you can have spherical or rod-shaped or spirals. So funny story, not funny at the time. Last semester, we were doing face-to-face labs for MOSO after a while, right? Because summer was completely remote and March, we kind of shut down suddenly. So things weren't necessarily put away the way we would have liked to. And so the lab had been closed for a while, and the water hadn't been used, the pure water hadn't been used, the incubators hadn't been open and closed. So there were multiple things that could have gone wrong. And I started to grow cells. Cells were having a hard time growing. Eventually they worked, they were working, they were growing. Students came in, they cultured the cells. And for the first time, I swear, for the first time in decades, I saw bacterial infection in the cells. And I'm like, oh my gosh, something, maybe the water is bad. Maybe the incubator needs to be cleaned again. It has been closed so long. But the odd thing was, usually if you have an infection, you just have a single type of infection. You just have spherical cells or rod cells. I had cultures where there were more type of bacteria growing in there than I could even think about. There were like 5 different shapes going in there. I'm like, what's going on? So we had to do a lot of problems, solving and a lot of cleanup before that all went away. It's gone now. I'm still kind of cleaning out all my old cultures and making sure everything is good. But now we are hypothesizing or thinking that some of this may be this COVID-19's fault. Why would I think that? Anyone have an answer? So, one thing is that obviously things were closed, so that could have caused some infections to float around. But if that was the case, I wouldn't have seen so many different types. So one of the things that we are thinking is that something is happening to the bacteria around us so that now they're surviving and they are growing in our cultures a lot more easily. Why do you think that may be the case?
Is it because, like, without intervention, a certain genera or species will like outcompete the others and will kind of like dominate it? Or
sure. Usually, if that happens, why does it dominate? Why is it that one type of species is surviving and growing in a certain environment?
It has some kind of resistance to whatever is affecting the others.
Yes. What are we using a lot of? So that's the funny part. What are we using a lot of nowadays?
Sanitizers?
Yes, we are using a lot of sanitizers nowadays. Now, we use antibiotics in our culture all the time to prevent infections. And that's fine, no problem. We don't get them. Like I said, first time I got so many cultures with infections in such a large variety. We are thinking that all the sanitizing use of hand sanitizers to the extent that we have done in the last year has actually done something to our bacteria in the environment. And we are going to see more antibiotic resistance and more resistant bacteria in general coming up now, because what else would have caused it, right? If one person may have caused an infection in a plate, I should have just seen that student's plate infected. I shouldn't have seen massive infections all over the place, and that too of various types. So just something to think about, that yes, we need to use them right now because we need to use them right now to kill other germs that we are trying not to get infected with. But we also have to be mindful of how it's affecting other organisms in our environment and how that may affect us down the road. And we have to think about the repercussions of those actions as well. So yeah, so I guess it's not a funny story. It's a scary story, but oh well. It was interesting. Okay, so here you have examples of some prokaryotic cells, and it shows how how different they can be in their environment. Here you can see that they do have, again, a plasma membrane and cytoplasmic space, and there is still stuff inside it. They may not have membrane-bound organelles, but they have molecules inside doing work that are needed for the survival of that cell. Okay, so prokaryotes are divided into 2 domains. You have your bacteria, obviously, and then the archaea. The biggest difference between those is going to be that bacteria typically live in normal environments, soil, water, humans, animals, plants, whatever. Archaea, however, can live in hostile environments. So they can live in acids, acidic environments, extreme cold, extreme heat. You can see them in Antarctica. You can see them in salt mines. You can see them in hot springs. That's the biggest difference inside them. And it is because of the way they metabolize and the way their membranes are structured that they can do that. On the other hand, your eukaryotes are going to have very clear compartmentalization, both on the outside and inside of the cell, to keep every separate function separate from each other. So they are kind of like mini multicellular organism in a way, if you think about it. So in an actual organism, a multicellular organism, your body is divided into compartments that do specific specialized function. In a eukaryotic cell, it's the same idea, but they are organelles that are divided, that it's divided into for specific specialized function. So cytosol is going to have a lot of different types of material that are going to be present inside. It's going to have the nucleus that has its own little membrane and housing the instructions of the cell inside. It's going to have the endoplasmic reticulum to make all the organelles and move them around through the Golgi apparatus. It's going to have the mitochondria to make energy. It's going to have lysosome for cleanup. All kinds of things will be present inside that cytosol. How much time do we have? Does anyone know? So I can get to a stopping point. Oh yeah, we have about 9 minutes. So we're good. I think. Okay, so eukaryotic cells are going to be, like I said, a lot more elaborate and a lot more involved than a simple prokaryotic cell. You should be able to identify and describe the function of each one of the organelles that are listed on this slide. Much of this is going to be described in the following slides anyway. So the nucleus is where you have your information for how to make the cell. The DNA inside, which is your information unit, that's your instruction book, is going to be packaged. It's long linear molecule, but it's going to be packaged into chromosomes and then repackaged with the help of helper proteins called histones, condensed inside the nucleus so that all of it can be packed in that little tight box. It's kind of like those, you know, the pop-up tunnels that you buy for kids, and then you can never put them back where they came from, because once they're out, it like fills up. Except chromosomes know how to do it. They can recondense and go back in and come back out as needed. Then you have your mitochondria that make your usable energy. They take your food, provide you with energy in the form of ATP that you can use to do all the work that you need to do to survive, to live, to work. And you have chloroplasts that are specific for plant cells that allow the plants to, without taking in any actual food, capture energy from sunlight, use the water and carbon dioxide to make their own food. And then once they've made it, they use it with the help of mitochondria to create energy as well. So the chloroplasts are these green organelles inside the structure, and that is what you're going to have. The chlorophyll is going to be these thin membranes inside the chloroplast that will allow for packing of sunlight. So these are like little solar cells. So a cool theory that we have is that mitochondria and chloroplasts are most likely long ago were most likely bacteria that got engulfed inside a predator cell that was an early eukaryote, right? An early eukaryote that could live without oxygen, obviously. So it was an anaerobic eukaryote that, as it was going about doing its eating of little organisms, ate a prokaryote, ate mitochondria that allowed it to become aerobic because it could still replicate and divide inside. And in the case of chloroplast, it then engulfed and ate a chloroplast that allowed it to become a plant cell that could make its own food as well. So the animals versus plant cells, the biggest difference that you will see is the presence of a cell wall on the outside, a presence of chloroplast in the inside in plant cells, right? And then presence of large vacuoles in the plant cell that maintain its structure and integrity. In the animal cell, it's not like they don't have any vacuoles. They do get vacuoles, but they're going to be a lot smaller and they're not going to be as large, and their functions are very different than what they are inside animals in a plant cell. In endoplasmic reticulum, you basically have lots and lots of membrane stacks, essentially, that are providing large surface area inside them, right, for different organelles to get assembled, for proteins to get assembled and modified so that they can do the work they need to do. They're usually going to be continuous with the nuclear membrane and the nuclear envelope. In addition, you have Golgi apparatus that are similar stacks of membranes, and inside spaces of these are going to hold proteins and molecules that need to get transported to other parts of the body as they are modified, and so they are packaged and moved to the different parts in the cell so that they can do their function, either through these vesicles that are budded off and carry them, or through another mechanism. Finally, you have your cytoskeletal structure. That is another way that cells can transport, not only hold a structure, but also transport material from one place to another inside the cell with the help of the microtubules and the tubulin and actin fibers that allow for movement of proteins and molecules from one place to another. Microtubules can also help the cells divide properly by helping to segregate the chromosomes in a dividing animal and plant cell. So that's what is going to create your spindle fibers needed for proper cell division to occur. The Golgi complex that we talked about earlier is part of the import-export system inside our cells that can both import materials through endocytosis from outside of the cell, as well as export material by exocytosis and throw it out of the cell as needed to maintain homeostasis and to communicate with cells around. Okay, any questions up till this point? One thing I realize is not part of my PowerPoint, but it is something very important for you guys to learn, is to be able to define the basic functions or basic things that define life. For example, the ability to replicate and divide, to grow, to interact with environment, all those things. I will make a special slide for it and post it in the module later today, because I do think that is a review that you do need in addition to the material that's here. So the last thing we're going to be talking about is model organisms. So in our, as molecular biologists and molecular and cellular biologists, we can't look at humans all the time for research purposes, because it is very hard to get humans to do what you want to do in a controlled manner. One, it's unethical in many cases to do the things that we would need to do in order to get the answers we want for our questions. And it's just not practical because of our general biology. We are very complex multicellular organisms with a very long lifespan. So it is not, and with very few progeny, very few offsprings. So we are not going to be the best organisms to research on in general, for general cell biology purposes. So instead, we focus on organisms that do have those properties that we need in order to do the work. Oh, what does the Golgi apparatus import and export again? Okay, so the Golgi apparatus will import and export proteins as their biggest thing. But it also takes macro and carbon dioxide, but it will take macromolecules or amino acids, like amino acids, nucleotides, things like that. It can transport as well. Majority of its function is to take signaling molecules in the form of proteins. It actually adds tags on them and different things as it moves them to the appropriate place, whether it is to the membrane or exporting it outside of the cell. When it's importing molecules, it's usually sugars or other like vitamin type molecules, amino acids that it's importing into the cell to take where it needs to go. Okay? Okay. So back to the model organisms. So for model organisms, like I said, molecular biologists and molecular biologists want something that has conservation, right, across genomes so that they can study a gene or a protein inside that organism and they can still relate it back to humans. So if we look at the genome, we see that there is a lot of similarity. There's a lot of conservation in the type of proteins and in the structures and functions and the genome sequences present all the way from bacteria to humans. So there are many, many proteins that are conserved. There are many pathways that are conserved that can easily be studied in a simple system like bacteria or yeast for eukaryotic purposes. And they can then be extrapolated to actual human systems and looked at the same way. For cell biology purposes, we want to look at structures that have, again, conservation at the genome level, but also at function level and in the general developmental side, the development of that organ the same way as it would be in humans. So for molecular biologists, we focus on E. coli or other bacteria, similar bacteria, to study basic pathways like DNA replication, DNA to protein translation, simple molecular pathways. For more complex genome analysis or more complex gene and proteins analysis, we use simple eukaryote like Cerevisiae, Saccharomyces cerevisiae, which is a simple brewer's yeast. Plants, typically for plant biology model organisms, many times they use Arabidopsis, which is a very simple weed plant that has simple structure that can be easily looked at, grow fast, lots of progeny, lots of kids, right? Lots of plants to look at, very simple, very easy. Same thing with E. coli and brewer's yeast, right? The same, you can get millions of actual organisms in a very short period of time to look at, to replicate, and to get your results very fast. For more complex developmental biology, we use flies, Drosophila, melanogaster, small tiny fruit flies. We use C. elegans, which are worms. We use zebrafish for development of vertebrate and mice for mammalian research. Those are model organisms for specific types of research in each case. Genetics is most advanced. We have all the genome information for all of these organisms. So it allows us to look at genes and proteins that are conserved and look at those pathways very easily. Again, Drosophila melanogaster, which are the flies, and the C. elegans, which are the worms, have lots of progeny to look at very quickly, have relatively short lifespan, so you can look at multiple generations and get large data very quickly. However, at the end of the day, we are not a fly, we are not a worm, we are not a mouse. We are humans, and so you do need to study those systems and replicate those findings in actual humans as well. And to do that, many times we do in vitro studies. In vitro literally means in glass, but essentially it is in a lab setting. So we do that by taking cells from humans, from various organs, from various systems, growing them in culture plates, growing them in flats, growing them on slides, and running our experiments in very controlled setting. We can also look at large human populations and look at the effect of particular types of mutations or diets in large human populations through epidemiological studies as well. Furthermore, we can compare the genome sequences from our model organism to our own and then look at common heritage between them and common pathways between them and extrapolate through that as well. So 2 words again that you would need to know from here, specifically for research purposes. In vitro which means in the lab, in glass, in cultured setting, and in vivo, which means in living system. So in actual living organism. So if we were doing a research in the flies, in the worms themselves, that's in vivo, or in actual human population, that's in vivo. If you're doing a experiment in cultured cell population outside the typical normal body environment, that would be what we would call in vitro. Okay, any questions on that? So I think we're running out of time. The next few slides are going to basically give you some reasons why we use each one of these model organisms. Some of it we've already kind of talked about as well, but that is something that you should think about, because that would be a very good question for you to answer on a test, for example. Being able to pick the right model organism for the type of research you want to do. Okay? Any questions?
No.
Everyone good? Chantel has gone to sleep. She hasn't gone to sleep, but she's tired. Was I that boring, Chantel?
No, I've been awake. I've been back-to-back classes all day, so
Oh,
since 8
okay.
a.m. I've been
Sorry.
You're good.
Well, you might want to take some coffee before your next class so your brain is fresh.
Yes, ma'am.
Okay, guys. I hope you guys were good. If you have any questions, if I went too fast, please do let me know so I can fix myself for next time. Okay, guys?
All right. Have a nice day.
You too. Take care, guys.
Thank you.
Thank
Have a
you.
good one, man.
Or Monday.
Bye-bye.
Depending on... Bye.
Thank you and have a good one.
You too. Take care.